Antibiotic resistance is a rising health concern, but how do diseases gain this resistance in the first place? Antibiotic resistance is the result of either a gene mutation or a gene transfer between a bacteria that gives them resistance to a specific drug. Mutations create new alleles from existing genes that provide resistance by interfering with the mechanisms of antibiotics. This can be done through a number of mechanisms like efflux pumps, alternative enzymes, target alteration, and more. These mechanisms work in a number of ways, some removing antibiotics from the bacterial cell, others preventing the antibiotics from entering the target destination. In the end, all mechanisms render the antibiotics useless against the bacteria in one way or another, causing bacteria with these mechanisms to have a survival advantage and evidently promoting resistance genes. Gene transfer is done by three methods: transduction, conjugation, and transformation, and it is the transfer of a resistance gene from one bacterium to another. Transduction uses phages, a virus that can transfer bacterial DNA from one cell to another. Conjugation is the physical connection of two bacterial cells that allows for gene transfer. Transformation is when one bacterial cell picks up genes that have been released from either a living or dead bacterial cell nearby. This is how resistance builds, but we still haven't answered the question of how bacteria are able to gain resistance. The answer to this question lies in the antibiotics themselves. Antibiotics create a pressure on the bacteria population to survive their effects, meaning that only bacteria with resistance genes are fit to survive in the presence of antibiotics. This is known as selective pressure. As the surviving resistant bacteria reproduce, more and more of the population will express these resistance genes, and the population evolves to be more resistant. Bacteria gain resistance independently of each other, so not every individual bacterium of the same species will be resistant to the same drugs. Further, some bacteria can develop resistance to multiple drugs at once. These are known as multi-drug-resistant bacteria, one of the most famous being methicillin-resistant Staphylococcus aureus or MRSA. MRSA is resistant to an entire class of normally efficient drugs, making it very dangerous and difficult to treat. So what can we do to avoid creating more resistant bacteria? Antibiotic use creates a selective pressure to evolve resistance, but this can be minimized by their proper use. Taking antibiotics when they are not needed unnecessarily exposes bacteria and creates the need for resistance. Healthy bacteria will be exposed to the antibiotic and killed off, leaving only those who have resistance to thrive and reproduce. This means that in the event of a future infection, these bacteria will already have resistance to antibiotics. It also means that there are more bacteria to share resistance genes with potential pathogens. Beginning antibiotics and not finishing them is another big issue, as the surviving bacteria have a chance at reproducing and creating resistant offspring. Not only will the survival of the bacteria prolong and worsen the infection, but it also gives the bacteria a chance to reproduce and gain resistance through genetic mutation. If this happens, the bacterial population will now be resistant to the same drug used to treat them the first time, and the infection will be more difficult to get rid of. The best way to ease the harm being done by antibiotic resistance is to educate the public and create awareness around the correct ways to take antibiotics responsibly, in order to minimize the emergence of resistant bacteria.